A very common sight, unfortunately, in a lot of rivers in the Midwest. This I think, is in the Missouri River, and these are silver carp, which is the main species that I will be talking about today. Oh, there we go. All right, and here we can see that silver carp have really taken over, and the big problem is the fact that they are very masterfully outcompeting native species. And I'm going to give you a little bit of insight today on what it is about their anatomy that allows for them to outcompete these other species. So there have been some perhaps creative solutions on how to deal with these silver carp. And so what you see here, hopefully you can see my pointer, this gentleman has nails driven through a bat that he actually goes out on a boat and tries to nail these fish. Another thing that's common, that has become common, is skiing while holding a machete and trying to get at these fishes, and also shooting arrows. People have been very badly hurt, jaws broken, in doing these sorts of activities. So yeah, that's not going to do much for controlling the population. So why is it that species become invasive? Oftentimes it's decreased competition. You generally have very few natural predators or competitors. There aren't parasites or diseases that are going to get you as much as the native fishes. You also have these fish communities where fishes become invasive. They are oftentimes very limited in diversity. The fishes that do become invasive have incredibly high reproductive rates, are very aggressive. If we can consider a fish having a personality, it's a very aggressive one. They are not scared of going into new environments, which is rather the problem. They tend to be very long-lived. They're usually generalists, so they can take advantage of just about any food available. And any disturbance within the ecosystem tends to increase that rate of invasion. So the impact of these Asian carp, they've already decimated many river systems, but the big fear is the fact that they are going to ultimately get into the Great Lakes region, and then we have a very big problem. They lay hundreds of thousands of eggs at a time, and as I said, they spread pretty easily into new habitats pretty quickly, pretty easily, and the cost of them invading the Great Lakes would be fairly catastrophic. Now, the thing that has allowed them to do so incredibly well is that they have managed to outcompete many of the native filter feeders because of the range of particles that they can consume. So when I'm talking about filter feeding, what are we talking about here? Fishes here, if we look into the gaping maw of this fish, you see here the gills, or the gill supports, are what fishes use in order to filter. Now, if we go ahead and take a closer look at what a gill looks like, these are the actual gill filaments here, the ones that look kind of orangish. That's where oxygen is coming from, or where they get oxygen into the body. However, they have on the other side these structures known as gill rakers. And these were long thought to work very much like a colander. When you're straining spaghetti, it was thought that the prey items would stick in these gill rakers, and this is the way they were able to catch food. As it turns out, it's actually much more complicated than that. So if we take a look here at these different fishes, and you see water coming in, this right here is a paddlefish. And this paddlefish is a fish that is a native species found with silver carp. However, they are being starved out because silver carp are so good at filter feeding that these paddlefishes that were considered to be the expert filter feeders actually can't get enough food. So I've been out sampling before, and we catch a paddlefish and it's absolutely emaciated because of the fact that it can't get food anywhere that there are lots of silver carp. Now, I had mentioned before that you have these gill rakers, and they were thought to act like a colander and strain food out, but it's actually not quite, it doesn't quite work that way. You actually have the hydrodynamics of food, particle-laden food, food-laden water actually comes in, and because of its interaction with these gill rakers, particularly if you see these really large gill rakers, they cause these localized vortices to form that tends to concentrate food. So that's what you're seeing here. Here are all of the food particles. As they pass over these gill rakers, they get quite concentrated. Okay, so this is what was considered, the paddlefish has long been considered to be one of the best, most efficient, effective filter feeders in the fish world. So what is going on with silver carp? What are they doing differently? So here is what a typical fish gill or the gill rakers look like. So here are the bony elements that hold those. Generally, we have one row of gill rakers, and you can tell from this fish how long those gill rakers are, that this is more than likely a filter feeding fish. Now, we look at big head carp. This is one of the invasive species. It is not the most dangerous one. The most dangerous one is the silver carp, but even the big head carp, you can see these very, very long, thin filaments that they use to, or rakers that they use to strain out particles. So they are very good filter feeders. But if we take a look at silver carp, we see something completely different here. So what you have here is not these individual gill rakers that together, very much like a comb, could be used to strain out particles. What you have here is a very solid wall of bone. So what's going on with that? If we take a look here, these are called scanning electron micrographs so that we can take a good look at the topography. This is what they look like just as a gross dissection. So again, this is the big head carp. It has the typical gill rakers, but here we have the silver carp. And you see, it doesn't look like any sort of filtering could occur here. And when we look at the scanning electron micrograph of this, you see that this is an incredibly complex structure, but one that doesn't immediately give the idea that this could be used for straining. So that was a question. Now, curiously, the development of this is very interesting because you see this is the earliest development right here at the very top. I trust that people can see my cursor moving around. I hope that's true. Bronwyn, is that true? What you see here are these individual gill rakers. And what happens during the course, as we're going down to the middle panel here, you can see a slightly older fish and even an older fish under that, is that these gill rakers become stitched together. So how exactly does that happen? If we look at a small, this is a larval silver carp, you see that they start out with these straight gill rakers. Here you can see a close-up. And even here at 25 millimeters, you still see that they are separate. They're sort of like this blade-like morphology, but there's no sort of stitching at all that is occurring. However, if we look at them when they are slightly older at 30 millimeters, what you see is that there is an increase in the length of each of the rakers. But really importantly, here, this arrowhead can show you, and here and on panel C, that arrowhead is showing that what we start to see in development is that those individual gill rakers start stitching themselves together. And this starts by actually the epithelium is what you're seeing here, stitching together. And then that epithelium becomes secondarily, it becomes ossified. So these are these little bony spurs that you see are helping to unite this. And this is a process that begins circa 30 millimeters, but if we follow that, you can see that by 60 and 80 millimeters, what you have are these complex plates. Here, all of these preparations are using something we called clearing and staining. So what you do is clear all of the connective soft tissue off and stain. The bone is red here, and then these bits of cartilage are blue. And then again, another scanning electron micrograph here that shows this increasing complexity of these plates. And here we have a preparation where we have snapped off a piece. So the outside bit is quite different in anatomy from the inside bit. But those two things working together is what allows for the filtration. So here we see a CT scan of a silver carp, and this blue is the entirety of that gill arch. And at this point is where the actual filtering occurs. And then this part up here is something I will get to ultimately. But you can see that a beautiful lattice is ultimately created in the adult. Now, if we take a look at this, again, the scanning electron micrographs here on the right and the cleared and stained images, and we look from the very top of this structure down to the bottom of this structure, you can see that there are these very interesting changes in the anatomy, which are going to be relevant in a little bit when I start talking about what this plate, this filtering plate actually does. But here you can see what the ultimate adult morphology is. Another thing that separates this silver carp from other species, the paddlefish that are very good filter feeders have only one row of gill rakers. Here you have these two plates, these two filtering plates. So why do you have two plates? And this takes me back to the mention of tongues. Here is where this comes in. So the palatal organ, as Bronwyn said, is this structure on the roof of the mouth. So here, what we have is this picture is showing you a head that has been bisected so that we are looking at the roof of the mouth here of this fish. And this is the palatal organ. Now, if we go ahead and take a slice through that palatal organ and see what the fish looks like, this is then doing a histological section so that we can see everything. This is the entire fish. This is a small goldfish. Here is this muscular pad, is this palatal organ. Here, for reference, are the gills all around. So what this palatal organ is this thick cushion of muscle on the roof of the mouth of many fishes. Up to 4,000 fishes within one group have this structure. But however, the function is really only well described in goldfish and carp, and not even silver carp, and we'll get into that in a little bit. But it's used to feed from the bottom. So how does that work? This palatal organ has the ability to make these protrusions that come down off of the palatal organ, and they can catch prey. Now, what this amounts to is this reflex loop. Because what you see right here up top is a very huge vagal lobe. This is the area of the brain that controls feeding in this species, in all of these species that have a palatal organ. And what happens is that there are a bunch of taste buds. The epithelium coating this palatal organ is absolutely studded with taste buds. That taste bud sends a signal that goes immediately to the brain and then back down to the palatal organ that says, that's a tasty morsel, hold on to it, and they're able to clamp down. The researcher who uncovered all of this and was a neurobiologist suggested this analogy. It's as if someone took a handful of gravel with one jelly bean in the center, popped it into their mouth, and within less than 1 second was able to capture the jelly bean and spit everything else out. This is so important in the life of the goldfish that the vagal lobe itself makes up 20 to 25% of the brain. So that is an incredibly important part of this feeding. So here is a very respectable palatal organ. This is in a sucker, another type of fish that has this structure. It's quite large. However, nothing really prepared me for the silver carp palatal organ. And here it is. Yes, this monstrosity is huge. Absolutely, on a big silver carp, this thing can probably weigh about ½ pound. And it has all of these different folds. You can see here 1, 2, 3, 4, 5, 6, 7, 8, 9 folds totally. What's going on there? And what's going on there is that this entire system is so closely related to each other. So those plates that I showed you, and I asked the question, why do you have 2 plates? The reason you have 2 plates is that one of these palatal folds actually goes and interdigitates into each of this pair of folds. Now, you could ask me, well, what does it do? And I wish I had an answer for you. We are not completely sure. It may be that it keeps the filtering plates clean. So you eat popcorn and you get annoying kernels stuck in your teeth and you're having at it with your tongue. It may be the exact same sort of thing where those palatal folds come in and keep that filtering plate very, very clean. It also can be that they piston pump up and down to help bring flow and to loosen the particles that have been collected. We don't know. I'm not sure that we will ever know. There are some big limitations to doing experiments with silver carp. But if we go ahead and try to get some clues from the anatomy, here is one of those folds. So we've pulled the fold out and done some histology. If we get in closer, you see all of these pink structures are muscles. And you see right along the edge here, they are all kinked up. This will tell you that those, when they stretch out fully, that palatal fold can be extended. So we know that they have to extend it, very much like an anteater tongue extends out. These folds, these palatal folds can extend down into those filtering plates. All right, so let's go back to the filtering plates. There are some serious issues with doing work on silver carp. Any place that is overrun by them is happy to send you 100 dead ones. They will not send you any live ones. So we cannot get data on live fish. So we have a very big constraint there. However, we have ways that we can model the system. So this is actually an SEM of the actual structure. This, on the other hand, is a CT scan of that filtering plate. So what you can do is actually 3D print out this structure and then test what effect all of this topography, this incredibly complex topography, has on water. And how do we do that? With this mouthful of a technique called particle image velocimetry. What does all of this mean? You can take, and here what we have mounted in this green is a laser sheet that goes through, but what you have here is a model that we 3D printed, and you put it in the middle of this tank of water, this flow tank, and water flows over it. And what we're able to do is see how different particles behave, because what you do is you have this water, you have it going at a set velocity, and then you seed that water with little particles. Those little particles will behave very much like the water or particles, food particles entrained in said water. So what we end up with, and I'm not going to go into the details of this here, there's a lot of some pretty heavy duty math and physics and hydrodynamics involved here, but the moral of the story is, as water passes over first this leading edge and then gets into these deeper valleys, so you see section 1 here and section 2 correspond to section 1 and 2 here, what happens is that you start having vortices of water formed. So just like, very much like a Dyson vacuum cleaner, where you can have that sort of these vortices being created, this is what allows particles to accumulate. So if you look here at D, what we're showing here is that filtering plate. And what happens is that water goes over and starts forming vortices, and then the particles are collected in those vortices and are dropped down, and they drop down in between here, and they fall to the bottom or are somehow moved to the bottom. Perhaps that's part of the palatal fold also. And then here we have this food transport groove, and that is what allows the food to actually be moved along. And as we determined from doing those PIV experiments, not only are these vortices formed here, but the direction of flow moves everything up into this area, which is this last area of interest, which is the epibranchial organ. This comes from another paper that published on this before, but they really did not do justice. Their epibranchial organs are these little teeny blue things. No, they're quite bigger than that. Here is an actual CT scan. This is sort of their interpretation. This is actually reality from a CT scan. And what you see here is that you have these epibranchial organs that are the ends of these filtering plates. So water goes over the filtering plates, prey is collected, and as I said, you have this food transport groove, and the food is moved along to the epibranchial organ. Now, what is that about? The problem, another problem that they have managed a great solution to, is if you are feeding on very, very small prey items, these fish, silver carp, can feed down to 8 microns. They can filter out bacteria and little accumulations of bacteria, phytoplankton, zooplankton, and they even have a way that they can not even send food through these filtering plates, just go bypass it completely and eat bigger things. So that's part of that generalized diet that makes for a really deadly invasive species. So let's get a little deeper into these epibranchial organs here. If we do this fish dissection and overlay, here is this big epibranchial organ. They also have, I didn't have time to get into this, but they also, here in this pink color, what you see is that they have pharyngeal jaws. So fishes have teeth on the oral jaws here. However, these fishes do not have teeth. but they have teeth in their throat. So this epibranchial organ helps to accumulate, aggregate, and concentrate food particles. And so they will ultimately move this into the esophagus. Now, there are other fishes that are filter feeders and have an epibranchial organ, and it's usually one tube like this. However, if you are really a very efficient feeder, you have a greatly expanded epibranchial organ, because here the tube is larger and larger, like a snail. It looks very much like a snail, and you have a greater surface area. They can accumulate more of this prey and then pass that along to the esophagus. So something that had an epibranchial organ like this would be considered an incredibly efficient feeder. However, silver carp actually have 4. So this is not only the most complex palatal organ that we have ever seen, it is also the most complex epibranchial organ that has ever been described in the literature. If we go ahead and take a cross-section through that, you can see these individual tubes here through cross-section, and everything that is surrounding these open tubes is muscle. So how this works is that all of these prey items are accumulated in the epibranchial organ. Then you have a big squeeze, like you squeeze a toothpaste tube, and all of that accumulated very small prey, but now it's a mass of prey, is sent off into the esophagus. So where have we been here? We see here the actual structure of these filtering plates. I talked a little bit about how they're different. I talked about the palatal organ and how those palatal folds fit into the gill rakers. I talked about the fact that we did, using models of these filtering plates, we know that it's this vortical structure that of the water and the particles that rotating allow for particles to be collected. And I ended finally with this epibranchial organ, and this is how prey are concentrated. So I didn't want to leave everyone hanging here. We have silver carp. They are a very large problem. How are we going to deal with this? If you can't beat them, eat them. And what we have is trying to develop a market. This has been very effective for lionfish in the Caribbean. Spearfishing lionfish has become a huge pastime of many scuba divers and even people who snorkel, and they actually are quite delicious. I've had them myself. The hope is to get folks to eat them. Unfortunately, they are very bony, and people don't like to mess with this. So thus the trying to convert them into fish burgers, and hopefully that will go. If we won't eat them, certainly someone else will. So there has now started a big export. Oddly, these silver carp got into our waterways because they were brought here to keep aquatic farms clean of algae, because they are such effective filter feeders. And what could ever happen? You know, they're contained. Well, they're contained until there's a hurricane. And then these fish manage with flooding to get released, and now we have a very big problem. But China and other Asian countries will happily eat them. I remember being at a talk I was giving at Boston College, and there was a Chinese postdoc who said, "Oh my God, now I know what is the part of the fish that we all fight over at the table, and it's the palatal organ, because it's very fatty and muscular, and so it really takes in flavor very nicely." I'm not sure that I could eat one, but I know too much. But anyway, we're hoping that we can start a market, and this can be really good for the United States and good for people who want to eat silver carp. Another thing that's been attempted is grinding them into fertilizer, although this then starts a problem because all of this fertilizer, the runoff of fertilizer into areas, is why we have some of the problems we do with algae becoming, these big algal blooms are due to the nitrogen from fertilizer. So we might be taking care of one problem and exacerbating some others with this. They have tried one final last resort is actually making pet food out of them. However, the stink of these pet food companies, I actually went to one of these to sample the silver carp they were bringing in, and they caused such a horrendous smell in the neighborhood that the neighborhood shut down the plant that was dealing with these fish. So there are some things in the works to get this problem under control, but it is still very much a problem. So with that, I would be happy to take any questions.
Wow, thank you so much. Let's unmute and we'll come back. I'll put a spotlight on you. Then I can open it up for questions. This was fascinating. Let me put a spotlight. There you go, spotlight on you. You can put a question in the chat, or you can raise your hand and I will call on you and you can unmute and ask Dr. Hernandez directly. Anybody have any questions? Yeah, go ahead, Pete.
Yeah. When I was in school at George Mason, I did a lot of work looking at pharyngeal teeth and minnows and other fish, and it was absolutely fascinating. And they let me use the electron microscope to get really good pictures. And it's amazing how big pharyngeal teeth are compared to the fish that they're in. I'm wondering, in these fish, do they have any purpose if you've already processed the food?
No, because you haven't really processed it in the epibranchial organ, that last stop before it gets I kept saying into the esophagus, but what it actually throws all of that mass, accumulated small particles into the area where the pharyngeal jaws are. And they have very flat, like a little brick like teeth. And what they do is these fish are just so bizarre. And I'm so glad someone actually knows what pharyngeal teeth are. So thank you for that. But they actually have lost the upper pharyngeal teeth. So usually you have upper and lower. They actually chew and process prey against the base of their skull. So they have this big keratinized pad on the bottom of their skull, and they have these teeth that work independently and just exactly like a mortar and pestle, because a lot of the things they're taking in are phytoplankton that have cell walls. And so they have to break those up. So very much like a mortar and pestle, they grind those down. And you can even see when you do scanning electron micrographs of the teeth, you can even see ridges on them from wear of breaking things down.
That's interesting. Thank you.
Mm-hmm.
I have a question. We had last week a discussion on eDNA and fisheries research here in Maryland, where they were taking the contents of the stomachs of the fish and seeing what they ate. Since they're such generalists, these carp, has any study been done on their preference? I mean, you said that they could eat big things, they could eat small things. Do they prefer big over small? Do they go to small if there's no big, or vice versa?
I think that in most cases, when you have that sort of generalist feeder, oftentimes they will go for whatever is available. So you know, they're not particularly choosy. I think based on the structure of the filtering plates, they have become very, very good at dealing with larger phytoplankton, smaller zooplankton. So things within that range. However, they can get down to 8 microns, which is incredibly small. And we have also found things that could not get through those filtering plates. So it had to get in via another route. So my guess is that they are probably marauding whatever area they happen to be in. And if they see a bunch of smallish shrimp, they're going to take advantage of those.
Okay. Are there any other fish that you found that has this type of gill structure, the gill raking structure that's been, ossified and pulled, stitched together, or is this just unique to this fish?
No, there are some other weirdos. The most of the weirdos are not bony fish, they're cartilaginous fish because, for example, manta rays, they filter huge things, but they're still filter feeding. They have some pretty complex filtering plates. But as far as bony fish, I think this is the most bizarre one, the most complex one.
Is this, there's a question just about the invasive catfish that's in Maryland. Is there anything that's comparable in terms of morphology?
They don't filter, but those are indeed generalist. Catfish, I have a friend who showed this ridiculous, well, frightening picture of a catfish actually eating a small armadillo. So, if you can get an armadillo down your maw, anything is open to you. So I think that that's one of those cases of, and I think with catfish also, their personalities are very, very aggressive. So they will go into an unknown area that many fish would like, it's scary, I'm not going in there. Boom, they'll go right in.
Other than just the industry of trying to create a market for the fish, is there are there any other ways to stop the fish from getting into other waterways? I mean, are we tracking it? Is it gonna keep going? Are we gonna get infected by them?
Well, I can tell you that they gave me a very hard no when I said, is there any way that we could get them in Maryland? It was like, absolutely not. Absolutely not. So to do experiments with them. There are some things they are using electrical. They're hoping that if they put these electrical gates in, the fish will start feeling a little buzzy and not go in, but they've already had them cross over. There was also an attempt, or at least a suggestion of, hey, why don't we put virus into the things they're feeding and then they'll eat the virus? And someone said, well, actually what they're eating is the natural food of both native species of fishes as well as mussels and other filter feeding mollusks. So that suggestion is not great, but they are indeed working on many different solutions here. A very frightening thing that just happened not that long ago was, I don't think it was silver carp, but it was a species related to silver carp. Eggs were consumed by something, defecated, and those eggs survived. So that's frightening. If you would ever have a fish of any variety that becomes invasive and then the eggs can actually survive going through the gut, then we've got big problems.
Has there been any silver lining? I mean, there was the silver lining with the zebra mussels that water quality is improved because of the filtering capacity, or are the other adverse effects completely outweigh any?
I think that any clearing up of water was sort of undone by the fact that the native species, and this is, paddlefish, even as adults, are filter feeding, but there are lots of really important sports fish that filter feed when they're younger and then switch. If they can't make it past that age, hard to see that one as a silver lining, unfortunately.
And then I thought it was completely fascinating, your ability to 3D print and 3D print and then model that in the tank. Had that been done previous,
Oh,
or
yeah.
was this
No,
the first study of its kind?
No. This for this question, it was the first time, but we have been doing all sorts, we, meaning scientists at large, have been doing all sorts of 3D printing for a variety of reasons. And the thing that's nice is that fluid being what it is, water being what it is, you can actually make it more viscous and you can change the hydrodynamic properties of it. So even things that are teeny tiny that are experiencing a different relative viscosity of the water, you can make a big model so that you can see what's happening and then just make the water really viscous. And then you've corrected for that. And then you have an appropriate scaled up experiment.
All right, somebody else had their hand up. It was Joe. Go ahead, Joe.
I haven't. Oh. I have an off the wall question.
I love those.
You showed the spiral associated. Hold one.
You had two mics going on at the same
Spiral
time. So there was
associated with the gills.
I'm sorry, I missed part of that. What was associated with the gills?
Just a minute.
Joe, you had two mics going on at the same.
The spiral.
Yeah.
Associated with with that gill structure.
Yes.
Is that spiral the same as the spiral that's in nautiluses that I think it's called a chi spiral, that mathematical formula that
Oh,
is found through nature in several different organisms?
yeah, I don't think so, but see, I love these questions that make me think. I'm going to go back and see if that's the case. But I don't know off the top of my head. Could be.
As a golden ratio kind of thing.
Yeah.
Yeah, whatever the term is, and I'm not I apologize for my lack of keeping that in my brain. But I know that there is that the Nautilus spiral is the same as in other animals that have that spiral with the snail shells and that it's a mathematical relationship that
Mhm.
that has that. I thought it was half a pi, something like that.
Yeah, no, I know what you're talking about, but I don't know if that would fit here, but I'll be looking into that. So thank
Okay.
you.
I was just wondering if that was something that was comparable across different species or certain type of species based on their characteristics and how they live and go through the food web and things of that nature.
Mm-hmm. Great. I'll look that up.
Thank you.
All right. Do we have any other questions for Dr. Hernandez tonight? Don't be shy. You got, okay. Well, thank you so much for sharing your time and your expertise with us. Everybody looks a lot smarter than they did when they first got on here, so you did a good job. Yep, Pete's got a thumbs up. Yeah, thank you so much for coming and sharing and all that you're doing to help us understand these and to solve some problems that we're facing now, and to keep them away from Maryland, hopefully.
Indeed. All right.
Deal with our own species. But thank you so much. Everybody
very
stay
much.
well.
Thank you, everyone.
Stay curious and stay outside, everybody. Bye-bye.